Hello. In today's video, we shall learn in detail about gene linkage. By the end of this video, you'll have a clear understanding about complete and incomplete linkage, coupling and repulsion hypothesis, Morgan's view of linkage with examples showing incomplete linkage in Drosophila and Maize. Let us begin with the introduction to gene linkage. Linkage and crossing over are the two main principles of inheritance and variation. Linkage may be defined as the nearness of genes or other DNA sequence to one another on the same chromosome, or the coexistence of two or more genes in the same chromosome is known as linkage. Genes present on the chromosomes have an inclination to inherit together. If two genes are located close to each other on a chromosome, greater will be the chances for them to be inherited together, or, in other words, they are less likely to experience recombination or crossing over. Linked genes tend to express together in groups from one generation to another generation. As the age of an individual advances, chances of crossing over decreases, resulting in increase of linkage. Rise in temperature helps in chiasmata formation and results in decrease in the strength of linkage. Next, let us look into the salient features of theory of linkage. Morgan and Castle formulated the theory of linkage. Genes that show linkage are located on the same pair of homologous chromosomes. Genes are arranged in a linear manner on the chromosome. Genes that are closely located show strong linkage. When two genes are present on the same chromosome, the chances of parental combination is much greater in the next generation when compared to the non-parental combination. Linked genes show two types of arrangements on the chromosome: cis arrangement and trans arrangement. In cis arrangement, the dominant allele of two or more pairs of linked genes are present on the same chromosome and their recessive alleles on the other homolog. In trans arrangement, a recessive and dominant allele for different traits are present on the same chromosome. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, let us study the different types of linkage. Depending on the presence or absence of new combination or non-parental combination, linkage is of two types: complete linkage and incomplete linkage. The phenomenon of inheritance of completely linked genes from the parental traits is known as complete linkage. The parental combination of traits are transmitted together for several generations. The linked genes, due to their closeness on the chromosome, do not separate and are inherited together. These linked genes do not show recombination and therefore do not produce non-parental combination. X chromosome of male Drosophila shows complete linkage. Since Y chromosome is without any allele of X-linked genes, pairing and crossing over does not take place. The incompletely linked genes are at greater distance from each other and have chances of separation by crossing over. The phenomenon of inheritance of incompletely linked genes is called incomplete linkage. They produce some percentage of non-parental combination along with the parental combination. In Zeer maize, the color and shape of grain show incomplete linkage. Next is Sutton's view of linkage. According to Sutton, genes are the units of heredity. Genes are present at specific points on the chromosome. During meiosis, genes present in the same chromosome will be linked together. Coupling and repulsion hypothesis was formulated by Bateson and Punnett. Coupling is the linkage of two dominant, two recessive alleles. The genes of identical parents enter the same gamete and are inherited together. A homozygous pea plant having dominant allele for blue flower and long pollen grains was crossed with another homozygous pea plant having recessive alleles for red flower and round pollen grains. The cross resulted in F1 individuals with blue flowers and long pollen grains. A test cross with F1 dihybrid with its double recessive parent showed phenotypic ratio of 7:1:1:7. The ratio shows that the dominant alleles and the recessive alleles have the tendency to remain together. This was termed as genetic coupling. In repulsion, the genes coming from different parents are separated during gamete formation and are inherited separately. The dominant alleles and recessive alleles of some genes are present on the same chromosome. A pea plant having dominant allele for blue flower and recessive allele for round pollen grains was crossed with another pea plant having recessive allele for red flower and dominant allele for long pollen grains. The cross resulted in F1 individuals with blue flower and long pollen grains. A test cross with F1 dihybrid with double recessive parent showed phenotypic ratio of 1:7:7:1. Now, let us understand Morgan's view on linkage. Morgan experimented with Drosophila and found that coupling and repulsion are the two aspects of linkage. Genes present in the same chromosome have a tendency to remain in their original combination and enter the same gamete. In heterozygous parent, the genes tend to enter different gametes and remain separated from each other. The strength of linkage depends upon the distance between the linked genes in the chromosome. The genes are linearly arranged in the chromosome. Some genes do not follow the principle of independent assortment. Let us learn about incomplete linkage in Drosophila. When genes are grouped on the same chromosome, some genes are tightly linked and show low recombination, while others are loosely linked and show high recombination. The genes for white-eyed and yellow body are tightly linked in Drosophila. When white-eyed yellow-bodied Drosophila was crossed with its wild type, 98.7% parental type combination and 1.3% recombination type were obtained. Percentage of parental combination was 62.8% when white-eyed miniature winged Drosophila was crossed with its wild type. Incomplete linkage is also seen in maize. Incomplete linkage between the alleles for color and shape of Zea maize seed was observed by Hutchinson. A cross between colored seed with full endosperm and colorless shrunken seed resulted in 96% parental combination and 4% new combination. So today we have learnt in detail about salient features and types of gene linkage, coupling and repulsion hypothesis, Morgan's view of linkage, incomplete linkage in Drosophila and Zea maize. I have some practice questions for you. Please share your answers in the comments section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.